Hey guys, and today we're going to be going over Edexcel AS Biology B revision, and we will be doing Carbohydrates 2, Polysaccharides. Okay. The most complex carbohydrates are the polysaccharides. They are made of many monosaccharide units joined by condensation reactions that form glycosidic bonds. Molecules with 3 to 10 sugar units are known as oligosaccharides, while molecules containing 11 or more monosaccharides are known as true polysaccharides. This won't really come up as anything you'd have to go through. Is just like telling you that a polysaccharide has to be more than 11 or more units, and so if it's just less than 11 but more than 2, it is a oligosaccharide. Polysaccharides do not have the sweet taste of many mono and disaccharides, but these complex polymers form some very important biological molecules. The structure of a polysaccharide makes them ideal as a storage molecule. They can form very compact molecules, so large numbers can be stored in a cell, so it's a concentrated storage. The glycosidic bonds are easily broken, allowing rapid release of monosaccharides units for cellular respiration, which is suitable for energy storage. They are not very soluble in water, so have little effect on water potential within a cell and cause no osmotic water movements. So they're not going to really interfere with cell processes, they're just there. The glycosidic bond between 2 monosaccharides is split by a process known as hydrolysis. So hydro is water and lysis is to split. So it's the opposite of condensation reaction, where we're going to put where from condensation we get a water molecule out. Here we're going to be adding a water molecule in for hydrolysis to occur. The hydrolysis reaction is the opposite of the condensation reaction that form the molecule, so water is added to the bond. Polysaccharides are gradually broken down into shorter and shorter chains, and eventually simple sugars are left, so monosaccharides. Disaccharides break down to form 2 monosaccharides. Hydrolysis takes place during digestion in the gut and also in the muscle and liver cells when the carbohydrate stores are broken down to release sugars for use in cellular respiration, because that's our storage. Our storage will be in the muscle and liver. And here is just showing you the process. So it is a reversible reaction. Carbohydrates as energy stores, so starch. Starch is particularly important as an energy store in plants. The sugars produced by photosynthesis are rapidly converted into starch, which is insoluble and compact, but can be broken down rapidly to release glucose when it is needed. Storage organs such as potatoes are particularly rich in starch, and so when you eat potatoes, they're not very sweet. And that we just saw in the beginning, and it told us that polysaccharides are not sweet. Starch is made up of long chains of alpha glucose, but if you look at it more closely, you will see that it is actually a mixture of 2 compounds. Okay, but just firstly, starch is the storage in plants only. So the storage in humans is something else that we will cover after. Amylose, so it's made of 2 compounds. Amylose is an unbranched polymer made up between 200 and 5,000 glucose molecules. As the chain lengthens, the molecule spirals, which makes it more compact for storage. So amylose, it's unbranched and it's got spirals, and that makes it compact for storage. Amylopectin is branched polymer of glucose molecules. The branching chains have many terminal glucose molecules that can be broken off rapidly when energy is needed. So terminal, so at the end. So it's branched and it's terminal and it's rapid. So here the spirals are good for compact storage, and here the terminals are good for rapid release. Amylose and amylopectin are both long chains of alpha glucose molecules. So why are the molecules so different? It all depends on the carbon atoms involved in the glycosidic bonds. Amylose is made up purely of alpha glucose molecules joined by 1,4 glycosidic bonds, which is why the molecules are long, unbranched chains. In amylopectin, many of the glucose molecules are joined by 1,4 glycosidic bonds, but there are also a few 1,6 glycosidic bonds. This results in branching chains that change the properties of the molecule. Yeah? So starch has a combination of straight chain amylose and branched chain amylopectin. So amylose is straight, so amylopectin is branched, and we saw that was the result of also 1,6 glycosylic bonds. This combination explains why carbohydrate foods like pasta are so good for you when you are doing sport. The amylopectin releases glucose for cellular respiration rapidly when needed, and amylose releases glucose more slowly over a longer period, keeping you going longer. Showing you that forms spirals, and here it is showing you that it is branched. Glycogen. Glycogen is sometimes referred to as animal starch because it is the only carbohydrate energy store found in animals, and that is the one that I told you is found in humans. And so where glycogen will be stored in the liver and muscle. It is also an important storage carbohydrate in fungi. Chemically, glycogen is very similar to the amylopectin molecules in starch and is also made up of many alpha-glucose units. Like starch, it is very compact, but the glycogen molecule has more 1,6 glycosidic bonds, giving it many side branches. As a result, glycogen can be broken down very rapidly. This makes it an ideal source of glucose for active tissues with a constantly high rate of cellular respiration, such as muscle and liver tissue. Because the muscle is always working, so is the liver. Carbohydrates in plants. Polysaccharides are very important in plants. Starch is the main energy storage material in plants. A typical starch grain in a plant contains 70 to 80% amylopectin, with the rest being amylose. I'm just showing you some pictures. That's in the liver, and the other one is in the plant cell. Cellulose is an important structural material in plants, the cell wall. So you might have come across cellulose during GCSE. But maybe not as a structure, but you've heard of it. So wall is an important feature that gives plants their strength and support. It is made up largely of insoluble cellulose. Cellulose has much in common with starch and glycogen. It consists of long chains of glucose joined by glycosidic bonds. However, as you will remember, there are two structural isomers of glucose, alpha glucose and beta glucose. In starch, the monomer units are alpha glucose. In cellulose, they are beta glucose and are held together by 1,4 glycosidic bonds, where one of the monomer units has to be turned around, so it's inverted, so the bonding takes place. So what they're telling you is basically here. As you can see, this beta glucose has switched. If you look at these, so actually it's flipped around, and that's because in order to make this bond occur, it cannot go like this. It would have to have been flipped over, like this. So the bonding can take place. The linking of beta glucose molecules means that the hydroxyl groups stick out on both sides of the molecule. This means hydrogen bonds can form between the partially positive charged hydrogen atoms of the hydroxyl groups and the partially negatively charged oxygen atoms in the other areas of the glucose molecules. Yeah, because we've talked about electronegativity, and so the OH, slightly negative, slightly positive, so it can form hydrogen bonds. This is known as cross-linking, and it holds together neighbouring chains firmly together. That's what they're talking about. Many of these hydrogen bonds form, making cellulose a material with considerable strength. Remember we said one hydrogen bond is weak, but many are strong? Cellulose molecules do not coil or spiral, they remain very long straight chains. In contrast, starch molecules with 1,4 and 1,6 glycosidic bonds between alpha glucose monomers form compact globular molecules that are useful for storage. So the shape is different. This difference in structure between starch and cellulose gives them very different properties and functions. Starch is an important source of energy in the diet for many animals. However, most animals do not possess the enzymes needed to break the 1,4-glycosidic bonds between the molecules of beta glucose, and so they cannot digest cellulose. So humans cannot digest cellulose, and that's why it's used as fiber, because it will pass straight out. Ruminants such as cows and sheep have bacteria, fungi and protozoa living in their gut which can produce cellulose digesting enzymes. It is the cellulose in plant food that acts as a roughage or fiber in the human gut diet, an important part of a healthy diet even though you cannot digest it.